So go ahead and get started on our extra module here. We've got about 20 minutes before lunch. It should be enough time, I think. So just as a reminder, every session of the workshop, including this one, is being live streamed and recorded. These presentations that are made today will go out on YouTube so that other groups can, or other hackathon participants can find you. And you've heard this 1,000 times by now, so I'm not going to go over it in too much detail. But I do want to post this hackathon folder link here. Give me just 1 second. So that if you want to pull up and look at anything that we're going to talk about here in a minute, you can do so. So here is the link to the hackathon folder. Should have everything you need in it. I'll post other links as we need them. And I guess for good measure, since we haven't posted in a little while, I'll post the Slack link again, just in the event that anybody in here has not yet joined the Slack. It is very useful for hackathon teaming. So just to go over some quick details of the hackathon, this is going to be review for some of you, but for those who haven't heard any of these presentations yet, we are having a 2-day model building hackathon at the end of the second week of the workshop. This will be taking place on Saturday, August 12th and Sunday, August 13th. We encourage people to work in teams for the hackathon. If you would really prefer to work alone, or if you can't find a group by the end of next week, it is okay to work alone, but we find that these projects typically are of the scale that you usually need a couple of teammates to help hack them into a workable state by the end of the weekend. These teams will be largely self-organizing, but if you're having trouble finding a team to join, or if you have a project idea and you're having trouble finding people to help you with it, we are available to help with teaming. And we'll have more opportunities to do that over the course of next week for those of you that are planning on attending the second week. What the hackathon looks like is you will have mostly large chunks of work time so that you and your group can work on your project as the instructors and helpers sort of circulate around to different groups to help get questions answered. We'll have check-ins with the whole group in the morning and at noon both days. And then in the second day, you will have, excuse me, grab a drink of water. Sorry about that. And then at the end of the second day, we'll have a sort of more formalized, it says written report here. You won't actually have to write anything up, but it'll be a more formalized summary presentation of how your group fared with your project, progress you've made, and what questions you still have. Because we do offer post-hackathon support for anybody who would like to continue working on their hackathon project. Or if you have another project you'd like to work on, maybe for a PhD thesis or for a publication, we'd be happy to continue to help you with those projects after the hackathon is over. So this is a good way, if you do have a project in mind that you would like to do in CompuCell, participating in the hackathon is a really good way to get your foot in the door with the CompuCell team for future support. We're always happy to help people after the workshop with whatever they may need, but if we're already familiar with your project from the hackathon, that kind of helping process is a lot easier because we've already worked with you pretty intensively for those 2 days. So if you do have a project in mind, hackathon's a good way to get our eyes on it early, and then even if it takes you a year or two to come back around to it, we'll still be familiar with it to some extent from the hackathon. These details are mostly the same. The one thing to highlight here is the second bullet point here. So we are going to have a few minutes today for a couple of lightning talk sessions. If you don't have time today for your talk, or if you have an idea that you're not quite ready to present yet, we will be doing 2 more of these on August 8th and August 10th. That's Tuesday and Thursday of next week. So you have some time over the weekend and the beginning of the week next week to get a quick project pitch together if you're interested in doing that. I will say, if you would like to participate in the hackathon, but you're not able to attend all of week 2 next week, that's perfectly fine. You are welcome just to come in for the extra sessions and give your project pitch. If you're not going to be able to make all of the sessions for that particular day, we know that the 2 weeks becomes a pretty intensive time investment. So you do not necessarily have to participate in all of next week to participate in the hackathon. We're perfectly welcome to come back, having only attended this week even, and still do your project. So there's no requirements there about attendance next week to participate in the hackathon. And we will be available intermittently over the course of next week to help scope project ideas into feasible CompuCell projects. So if you have an idea that you feel is a little bit too ambitious or too broad, we're happy to help you pick a piece of that that you can successfully implement in CompuCell. And we'll actually have a couple of talks in the extra sessions next week specifically about how to go about this process of scoping a project idea down into something achievable within the hackathon time limits. Couple of expectations. If you are planning on participating in the hackathon, we do like you to have some idea of what to model and why. If you don't have a specific project in mind, having some idea of what kinds of topics you are interested in working with is also helpful, so that we can help place you with a group a little bit easier. And one thing that is super useful is to have some kind of shareable description of yourself and your skill set or background, so that if you are looking for a group, a project lead can potentially reach out to you if they've seen your skill set and say, hey, listen, we think you'd be a good fit. Would you want to work with us? We have a shared teaming document, which I'll talk about in just a minute, where you can do some of that. But something else that you can easily send to people, say, hey, I'm good at this. What do you need in your group? Is very useful, so that all doesn't have to happen, verbally between all of the groups. Yes, thank you, James. James has just launched our hackathon participation survey. If you could take a minute to fill this out while I'm talking, that would be great. This is just to get a feeling of how many people want to participate in the hackathon and what level of or what the status of your project idea is if you have one, or if you're just looking to join another group. So please do fill out that poll. And then expectations you can have of us is obviously that we will be available throughout the course of the hackathon. And then, like I said, after the hackathon as well. Just checking the chat here real quick. One thing we do suggest is if you haven't done the Rosalind exercises that we recommend is a good way of sort of testing your Python skills. We do recommend that you take a stab at those. I'll post the link in the chat. If you can get through exercise 5 on Rosalind, you're in pretty good shape to participate in the hackathon. Now, again, this is not a requirement to participate in the hackathon, but if you are nervous about your Python skills, seeing if you can make your way through the 5th exercise on the Rosalind Python Training Village is a good way to determine, whether or not you're at the right place to start building your own models in the hackathon. Again, we can help you get there if you're not able to get through them. But like I said, if you can get through exercise 5, you should be set for the hackathon. And again, you don't have to be a coding expert. Groups are made up of a lot of different types of people. Not everybody in the team needs to be a Python wizard. As long as you have one person in the group that knows some level of programming, most groups are able to get through their project into a working prototype stage. So again, it's more about having at least somebody in your group that is okay with Python. Not everybody in the group needs to be able to get through the Rosalind exercises, for example, if that makes sense. So here's our links. Yes, like James says, with the poll here, there's no commitment now. But first of all, the poll is anonymous, so we don't know who's saying this anyway. But even if you do say you're interested in participating now and feel like you don't want to later, that's perfectly fine. If you say no when you change your mind later, we're obviously happy to, to bring you on to the hackathon Anyway, so again, no commitment here. This is just for us to get an internal sense of how many people to expect to plan for the hackathon, because we have to organize things a little bit differently depending on the scale of the participant pool. So let me post these here. So we have this hackathon teaming document, which I'll post the link to here. And we'll take a very quick look. I think 2 people put their hands up about lightning talk, so I do want to give time for that here. Basically, the way this document works is if you have an idea and you're looking for teammates, put your contact information here. The first line is the format of how to fill it out. We'll need your name, an email, and a descriptive project title. You can write more about your project in the pages below. But if you're looking for a project to join, you'll need to put your name, email, a couple of research interests of things that you're interested in working on, as well as, like I was saying before, a short list of relevant skills that you think would be useful to a hackathon project group. For now, you can ignore requested breakout room names. That's for once we establish groups within the hackathon. Once we have our teams set. So in the pages following that, we have a template here for people to add their project idea. So what you'll do is you'll create a new page at the bottom of the document, copy and paste these headers in, and then fill these out with title of the project, brief description, what you're looking for in potential partners. I think we had a question about this last time, James, about what the appropriate models to use is. So if this is for resources, if you have resources that you'd like to share with people, so if maybe there's a publication that you're basing your project on, you would put that here. Or if it's something that hasn't been done yet, you would need to provide a little bit of background information.
Not sharing the doc, your screen sharing the teaming.
Okay. Sorry about that. All right, so let me run over that again real quick. Is it sharing now?
That's fine.
Okay. So like I was saying, if you're looking for a project group, or sorry, if you have a project idea, you would put your name in this area. Again, there's a template to follow here. If you're looking for projects to join, list your name here. Here's the template for that. And then, like I was saying, if you have a project idea, you would make a second page or another page at the end of the document, copy this template in and fill out each of these prompts, except for organizers' comments. That is for us to come in over the course of next week and leave some feedback on various aspects of the project and things that we think would be important to know moving forward as you create your teams and prepare for the hackathon. Yes, thank you, Wilson. James makes a good point that it might be useful to show a hackathon project from last year. I don't know if we have anybody in here currently. I know Karina was in here this morning. Maybe we can recruit somebody tomorrow to show their hackathon results. Oh, Karina is still here. So again, no pressure here, but if anybody in the room participated in the hackathon last year and would be interested in presenting basically the results of their hackathon project either tomorrow or maybe on Monday, we can talk about that. If not, I can run through one of them for us tomorrow. Because I think we should still have access to most of those files. All right, is it showing the PowerPoint again?
It's fine. You're back on the PowerPoint.
Okay. All right, we'll talk a little bit more about the schedule in one of the next meetings. One other thing that I do want to mention is we do have this hackathon project ideas document. And again, a lot of you have heard about this already. Let me go ahead and grab it. One thing that we think is useful if you would like to participate in the hackathon, but you're not really sure what you want to do, is to try to reproduce a model from a published paper that hasn't been done in Compuso. That takes away some of the conceptual heavy lifting for you up front. And so you can focus mainly on the implementation phase, which is one of the hard parts, particularly when you only have 2 days to do it. So if you have any questions about this document or need help narrowing down potential projects from this list, please do email me and we can talk about that after the fact. So I know I had at least one hand up during lightning talks. I think it was Cesar. Yes. Cesar, would you still like to present today? We've got just about 3 minutes, which is actually the target time for these things. So, and then Karina, were you saying you wanted to present today too as well? So Cesar, yeah, so Cesar needs to be unmuted. Let me do that. What about now, Cesar? Can you unmute now? There we go. So we'll have Cesar go first, and then we may need to run over a couple of minutes. Karina, we can have you do at least a short rundown, and then if you want to represent next week, that's fine.
Hi, can you hear me?
Yes, here you go. Awesome. You want to go ahead and screen share?
Let me. Okay, so can everybody see the presentation? I'm sorry, I'm not seeing the. Okay, so I'll very quickly present my project. This is my PhD project. It's called Misconstruction in the multicellular development of Mixococcus santos. This is a bacterial species that is known for being able to develop into multicellular structures called fruiting bodies during their life cycle. And these bacteria usually are in, how do you say, well, they are usually spread out in the medium, and when there's some lack of nutrients stress, they start to aggregate. And during this process of aggregation, they actually leave modifications or modify their environment in ways that allows them to migrate collectively and to form these aggregates. So that's the entirety of my project, to study the impact of these environmental modifications in from a niche constructing scope. Niche construction is just a term used to describe processes of environmental modifications that have evolutionary consequences. And so in these two images, we can see a set of a group of blue dots and yellow lines. The blue dots represent cells, well, are marked cells, and the yellow lines are trails that they leave behind. And we found that, or we think that most of the environmental modifications that Misconstruction induce during their aggregation are limited to these trails, and the process of they follow these trails like roads, and they change their behavior when they're within these trails. So these are two pictures taken 12 and 36 hours respectively within the multicellular development process. Again, niche construction is refers to these environmental modifications that have an evolutionary impact. And the idea is that, well, usually when people study the evolutionary transition to multicellularity, the approach is more of an adaptative approach, where the explanations revolve around how multicellularity has adaptative benefits to the organisms, but they leave behind explanations regarding the bidirectionality of the relation between the organism and the environment. So the idea is that environmental modifications, induced environmental modifications by microorganisms in the past, were determinant for this evolutionary transition. And so, well, answering very quickly the questions of the template. Again, this, at least in our current setup in the lab, we have problems putting to test how and which modifications are in play when the cells aggregate, when the bacteria start to aggregate. And so quantitative model would help us to approximate explanations in this regard. And we are trying to see initially if there's a threshold of specific environmental modifications that allow for multicellular development, or prevent it if there isn't that threshold, if that threshold isn't met. And when we think that a critical degree of environmental modifications will prevent or allow the aggregation and multicellular development of these organisms. I didn't answer some of the questions because I wasn't sure how, what to say about them. But well, the Compusol is an excellent platform for this because, again, the biological system we're studying, that is Misconstruction, is a model for multicellularity and, sort of speak, tissue development. And these fruiting bodies are multicellular, not just because they're made out of many cells, but also because there's specialization within those cells. They differentiate in different cellular types. And yeah, we expect to see different phenotypes and migration dynamics, and more than that, we want to approximate an explanation on how that would be translated in real life, in the real life model, where we can eventually modulate specific conditions in the wet lab and yeah, prove that what we are modeling is something that actually happens. And so, well, a very basic project outline. At first, I don't expect to have a very complex model at this point, because at the end of the hackathon, because I myself know nothing about any of this. but I'm thinking about first modeling bacterial cells, both within that are realistic dynamics and like body types, that they move like bacteria essentially and look like bacteria. And after that, well, to model the trill secretion and some properties of those trills, then as the more bacteria that passes through a trail or a section of the medium, the bigger the trail will become, and that it degrades over time. And then model the cell-trail interaction, where cells prefer to move along trails and have differences in speeds and other things when they're in them. And well, here I have a few articles, 3 that are very, very basic for all of this that I can show you there. And well, my background, I'm a biologist. I prefer to say that I have no skills in computational modeling, and I'm completely new to this. So I don't expect that I will be of much help. At least, I don't know. I think that the model I want to develop is also one of the training exercise for me. And yeah, I don't know, anybody that who is interested would be awesome to have. And yeah, I'll stop sharing.
Okay, great. Thank you. And like we said, it's perfectly fine to just come in as a biologist. Typically, these teams need somebody with an understanding of the biological problem to organize the group and scope the project and everything like that. So that's perfectly fine. James,
a couple of papers, mainly the papers that I'm finding about Myxobacteria, which are actually done by Yi Zhang, who uses CompuCell, but they didn't use CompuCell. But I do find a couple of Myxobacterial papers that seem to use CompuCell. So those might be a place to start if you haven't worked with them, if those aren't papers you already have.
Yes, I actually have some papers. In fact, in my lab, CompuCell has been used to model Myxobacteria. It's just that I've been I've been working so much in the wet lab that that's why I said I prefer to say I know nothing about this, because I don't know how much I do and I do not know. But yeah.
But certainly, it's fine for a hackathon. It's fine for a hackathon project. I'll say that again. It's fine for a hackathon project to take an existing model and extend it. So if there's a lab model that you want to dig into and extend with other people, that would be fine. If you want to start from scratch, that's fine too. But if there's a published one from your lab that you want to dig into for 2 days, so we'd be happy to help you with it.
Thank you.
all right, so Karina, I think, wanted to present quickly as well, and then we will go to lunch, and we can come back from lunch a little bit later since we've run into people's lunchtime. But Karina does want to present very quickly in case there's anybody here who wasn't planning on coming to week 2 but may be interested in the project. So Karina, let me ask you to unmute. Yep, there you go.
Yes, I hope you can hear me now.
Yes.
Great. Let me briefly present my work. Can you see the slides right now?
Yeah, it looks
You
good.
see my screen? Okay, good, because I cannot see the Zoom anymore. So I want to pitch an idea for actually creating a demo for extracellular matrix with like a fiber structure to study like this interaction and maybe the dynamics that, well, emerges from the fact that there's extracellular matrix. So why would you want to model the extracellular matrix? Well, it plays an important role in cell migration, and there are like a lot of aspects to think about. So like all these things matter. So for example, in what way is your matrix structured? What's the stiffness of your matrix? Maybe there are some gradients, there might be some different components in there, and all these different components can determine how your cells migrate. And I think some of these aspects can quite nicely be modeled in the cellular pods model. So I think it would be nice to explore how this is done. And well, there are a lot of different cellular pods model papers that incorporate fibers. So I just tried to collect a few. So here you have like random fibers and you have a little small tumor in the middle, and they try to see how does these cells start to migrate, but also how do cells migrate on a very regular structure. And well, I can pop in some more examples where you have like 2D versus 3D. Something here with diffusion fields where these cells secrete something, and this chemical actually, how do you say this? Like destroys the network, so it opens up new spaces. And they also tried, for example, to see like what do different kind of geometries do to the migration of cells. So there's like a lot of things in literature, but one thing that I think would be cool is just to have a simple demo where you have some cells and some fibers, and where you can just play with a lot of the functionalities of CompuCell 3D. So yeah, I think that would be something nice to explore in 2 days. So in the end, it would be nice to have a demo which cell and extracellular matrix interactions can be explored. So what I was thinking of, like a time planning for those 2 days, maybe in the first day we can like focus on how to represent the extracellular matrix with fibers and cells. And then on the second day, explore a bit of different kinds of functionalities depending on what you're interested in. So that at the end of day 1, we have like a basic model, and on the next day, we can either individually or together explore different things that we think could be interesting. So maybe we want to look at diffusion fields or adhesion, or we want to look at like explicit migration. Well, I'm not entirely sure, but I think we can think of something nice to show that such a demo is very useful. So about me, I'm a PhD student, and I have been using both CompuCell and the cellular pods model now for over a year. And I think anyone who's interested in like thinking about cell ECM fiber interactions is very welcome to join the project. If you just want to try out some functionalities in this biological context, you're very welcome to join. And also, if you really like to work on making a very simple model that can be beneficial for others to like further think about this topic. So yeah, I think that was my presentation very briefly.
Great,
Yes.
thank you. Any quick comments, James, before we break for lunch?
I was typing some of them in the chat. I wanted to see this happen for years. So I'm delighted if we could do it. Of course, Pauline Hogeveg already had some nice fiber models 25 years ago, which we never pulled into CompuCell. It would be great to see that. Of course, Roland has done those as well. I think the issues of how do fibers remodel when cells move past them, and how do you model the biasing of cell movement along the direction of fibers. I mean, in principle, if you have, for example, the fibers represented as a vector field, then when you do a copy, you want the copy probability to depend on the relative orientation of the fibers and the copy direction. So you do a dot product there. And
yeah, another thing I was thinking of is like having a single layer of fibers and then a layer of cells on top of them, such that they fill each other and they can like interact with adhesions, but not being blocked, because that's really obviously a 2D problem. But yeah, I'm not entirely sure how to model this, so I thought it might be nice to explore just in the weekends.
So Yi has done it with explicit fibers, where the fibers actually are a cell type.
Yeah.
That works fine, and people like Andreas Deutsch have done that. And then other people like Pauline and Roland have done it with fields, where the fibers are represented as a vector field, where the local orientation of the fiber is represented by the field direction, and the degree of orientation of the density is represented by the amplitude. Both of those work. I think to do the field cell interaction, to get the cells to move preferentially along the fiber architecture direction, ideally you'd want to write that in a C++ plugin, but you could dry run it in Python. And then once you have an algorithm that works, then we could see about having one of our team implement that in C++ to speed it up. So I think that's a great project. They're just sort of pulling together all the different fiber models and how they work, and coming up with one or two. But just the idea that when the cell moves along the fiber, it tends to reorient the fiber in the direction of movement. And the idea that the cell preferentially moves along the fiber direction, implementing those two things. So the idea of combing of fibers and bundling of fibers already is very, very powerful. And that can be implemented, I think, pretty simply. TJ, I hope, will be around, and he's certainly worked on that a bit in his thesis. So I think there's some really good opportunities there. I'd love to see that project come to fruition. I'd love to see somebody who's a good C++ programmer join you, so we can actually capture that stuff inside of core contours.
Yeah, that would be nice.